Okay, Nick.
Okay, so I make that just on the hour, so I think time to start. And while I introduce the webinar, then we'll have a few more people coming in, I'm sure. So welcome everyone, and thank you for joining us in what is the last instalment of the biosimilars analysis series. My name is Nick Pittman. I'm the global marketing manager for Biopharma at Waters. It's my pleasure to be your host today. The last episode in the series has come around pretty quickly. But just to quickly recap on some of the previous sessions, we started off a few weeks ago now, and we heard Andrew talking about some chromatographic techniques for characterizing critical quality attributes in biosimilars. Then we moved on to episode 2, where Sam talked us through some really nice workflows for biosimilar comparability assessment. And just last week, Pavel discussed some nice approaches for LC method development in the context of biosimilar analysis. All of the above sessions were recorded. Those recordings are available on demand, and we're going to send out some links for the whole series. They're going to be shared after this particular event, so keep an eye out for those. Now, as I say, this week is our final week. We are delighted to have Jared Young joining us from Similis Bio to talk about a collaboration that we've been working on between our two companies on some subunit analysis for monitoring multiple attributes of biosimilars during bioprocessing and development. Jared, if you'd move us on to the next slide, please. It'd be my pleasure to introduce you. So Jared is an analytical scientist. He has a combination of scientific expertise and also computer science skills. His degree is in chemical and biological engineering from the University of Colorado, and his industrial experience began in Amgen's biophysical characterization group, and he then became a founding member of Elion Labs, who were later acquired by KBI. Where he served as a director in analytical and formulation sciences. Now, Jared was an inaugural member of Similis Bio, which launched in 2022, where as a director of biosimilar strategy, he leads analytical method development and reference product characterization. When he's not working, and it sounds like he's working quite a lot of the time, but when he's not working in his spare time, Jared can be found in the great Colorado outdoors, either hiking or skiing with his family, both of which sound pretty awesome. So I'm a bit jealous about that. Okay, if we can move on to the next slide, please, Jared. Just a couple of very quick points on today's Zoom platform. You'll notice, first of all, that your cameras and your microphones have been switched off as a default, but that doesn't mean we don't want to hear from you. We always love your questions and comments, so please do ask anything you would like in the Q&A box, which you'll see on your screens. We'll address all of your questions at the end of Jared's presentation. And just to note that this session, like the others, is being recorded, and a reminder that we'll share those recordings after the session. Right, that's more than enough from me. I'd like to welcome Jared again and ask you to begin your talk.
Thank you very much, Nick. I appreciate the kind introduction. As Nick already said, today I'll be talking about streamlined antibody subunit LCMS workflow for monitoring multiple attributes during antibody candidate selection. So this is early on in the process, clone selection, and then also this method has applications in early upstream process development. But first, I'll start with a little bit of an introduction about Similis Bio, what we do, and try and kind of lay the context for why we were interested in this particular approach. So the origin of Similis Bio, as Nick mentioned, my last position was at KBI Biopharma. We were, I was in the analytical group there, so we would work with multiple biosimilar companies. And the current biosimilar paradigm, each company goes out and sources their own reference product, develops analytical methods to characterize those reference products, and then goes on and performs the same analytical testing on each biosimilar product independently. And each company has siloed data sets and siloed expertise in each of those analytical methods. And as a contract analytical provider, we would see the same requests coming through over and over again for the same biosimilar, or for the same molecules. And so what we wanted to do with Similis Bio was eliminate the waste that we were seeing in the analytical space by independently sourcing the reference product, developing analytical methods, and putting together data packages and analytical methods, and then selling those data and methods rather than selling services to recreate the same methods and data over and over again. So the idea was to eliminate waste, reduce development costs, and also improve quality, because by reducing waste, you can invest more time and effort and money into developing high quality methods and high quality data packages, which can be, sold at lower cost than doing the services directly if we sell them multiple times. So that was where we started, very much an analytical origin for Similis Bio. The scope of what we do quickly grew because there are many other components that go into the decision-making process in terms of what programs we're going to target. So patent discovery came up. That's something that for each biosimilar program, we do detailed patent discovery work with multiple different organizations to find as many patents as we can, which are, as you can imagine, quite numerous, as well as analyzing key patents, both in terms of freedom to operate, process patents, verifying when we think those products will hit the market. Analytical development, which I've discussed, but quickly received requests for asset development as well. So these would be cell lines, and then upstream and downstream processes. So Similis Bio, if I didn't mention this, is a JSR Life Sciences company. So we're in the family with Celexus, which is now KBI, and KBI Biopharma, which are CDMO. And so we have access to those capabilities. And so we actually have begun our own cell line development and upstream and downstream process development as well, and moving those all the way forward to tox lot production. So yeah, that's what Similis does. Give an idea of the scope of the work that we do. We work on a lot of products. So here's the list of, it's actually not a complete list, but the list of programs that we've moved forward kind of past the initial evaluation phase, where we actually have analytical data and methods. And it's quite extensive. So, you can see we have, I don't know what that is, roughly 20 there. We've sourced, between 30 and 40 products. We have a large set of analytical methods and excuse me, data on each of these products. And yeah, I guess I'll be brief on this just to get, the main takeaway here is the scope of what we're doing is quite large. And in terms of cell line and process development, that leads to a very large number of cell lines and clones that we're evaluating in order to, figure out commercially viable candidates to move forward into, from transfection to clone selection and into upstream process development. So here we've got 25 different ongoing programs. And so the scope of the number of samples that we're testing every day is quite large. So what we collaborated with Waters to do was to develop a LC-MS method to look at the key attributes that we look at during clone selection. So this is post-transfection. We've got several single cell clones that we're looking, at RCP state, looking to pick the top clone based off of productivity as well as the quality attribute profile, which is going to be key for biosimilar. And the two main quality attributes that are inherently heterogeneous and have kind of different base profiles at the clone stage and at cell line, and looking at different cell lines, are the charge profile and the glycan profile. What the figure here shown on the left is a ICIEF trace. So we've got our acidic, main, and basic variants. And what's being shown here is a CHOM clone or an SP2 clone. So that in this particular case, we've got the SP20 innovator, which has higher levels of basic species. We expect these to be C-terminal lysine variants. But before you start a $100 million program, you need to confirm that these actually are C-terminal lysine variants. And then the other charge variants in the acidic region can vary a little bit more, but again, we need to verify what those are. And as you can see, in this particular case, switching cell lines leads to a fairly substantial difference in the charge profile. And then we've evaluated 3 clones, for each of these. For the CHOM cell line, and we get some idea of the variability of each of those acidic species. And then so that's the charge profile, which is important for biosimilar clone evaluation. I would say more importantly, or what we tend to look at more closely for clone selection is the glycan profile, because this has typically what we'll see with antibodies is, C-terminal lysine variants are the big driver there, which don't have an impact on safety and efficacy because they're quickly cleaved in vivo. But the glycan profile does have a fairly substantial impact on PK. So that, in particular, if there's no effector function, your high mannose, aglycosylated, and sialic acid are going to impact clearance rates, as well as aggregation. And so, we look at those very closely if there's no effector function for antibodies. And then if there is effector function, then the level of fucosylation is going to be critical, because that can, it can impact your ADCC activity, orders of magnitude. So that needs to be spot on for analytical similarity for biosimilar. And then level of galactosylation as well can impact your ADCC activity, twofold or so. But again, just to recap, we are evaluating a ton of programs. We're looking at, for each program, we're looking at tens to, we haven't it narrowed down, maybe 4 top clones that we're looking to move forward. And then, for charge variant characterization and glycan characterization, the scope of samples tested and analytical, just the number of samples tested is quite large. So collaborated with Waters to develop a single scalable method to monitor the attributes that we cared about, which again, charge profile and glycan profile. And so to give an overview of how that method works, the initial study that we ran used a Ambr 250 material. So we have 2 different, excuse me, 2 different clones, and actually 2 different cell culture conditions as well, and innovator material run alongside with that material. That was clarified cell harvest. So the sample prep needed to be automated in order to do a Pro-A purification. Excuse me. Do the Pro-A purification so that we could analyze this by the LC method. The antibodies were digested by Fabricator or IDES and reduced to get them into, the FC light chain, and then FD, which is the Fab domain of the heavy chain. And then those were loaded, run by reverse phase method, and analyzed on the BioAccord system. And then data was processed using Waters software, and then we were able to integrate relative abundance of the tick traces to get levels of relative abundance of galactosylation, C-terminal lysine, glycation, and oxidation. And then these results were ultimately compared back to what we were previously using as our ground truth, which was the Waters Rapafluor method, which is a fluorescence-based glycan method, and ICIEF data for the charge variants. So for the sample, so that's kind of the general overview. I'll go through each of the different steps of the method, kind of in order. So the first step is sample preparation, obviously. So this was done using the Andrew Plus pipetting robot. It had the extraction plus and heat shaker modules on it. So all of these steps could be automated. I wasn't very familiar with the Andrew Plus robot before this study, but researching that and learning about its capabilities is pretty exciting. It uses, standard Sartorius pipettes, but you can easily automate the workflow to make it so that the analyst interaction is minimal from beginning to end. So the protein A purification used the Cytiva MabSelect resin on collection plates. There was a shaker step that could be automated. This was run multiple times, so I don't think it was automated every single run, but again, that could be automated. And then the Fabricator master makes preparation. The preparation was done in an automated fashion, but the enzyme was added manually just to conserve the amount of enzyme that was used. And then after the Pro-A purification of the clarified cell harvest was done, the samples were added with were combined with the master mix and Fabricator enzyme. They were incubated at 37 degrees Celsius for 30 minutes. And then reduced and diluted for analysis on by the LCMS method. So that was done. Yeah. And am I missing anything here? Don't think so. Here are the parameters for the LCMS method. So this was run on an Acuity Premier UPLC with UV absorption at 280 nanometers. There was a polyphenol reverse phase column used. You can see the flow rates and gradient there. Is a fairly short 15-minute method, which again, for large sample volume is important to keep the method short. The mass spec was done with the QD RDA, positive ionization mode. Don't need to read all those off. You can read them, but those are the parameters used there. Overall, the method is, I think, fairly straightforward on the LC and MS side. The BioAccord system is designed to be easy to set up, easy to calibrate, relatively straightforward to run. So proof true here. For the data processing, I guess here are the UV traces and tick traces, I apologize, here on the left. So we get 3 peaks as expected. So the first peak is the FC, and then we have the light chain, and then the FD, or the cleaved, the fab portion of that heavy chain. The data that the mass spec data shown on the right is just for the FC domain. So we have and this is the glycosylated domain. So we have MAN5, G0, G0F labeled here. And then we also have C-terminal lysine, which is our primary expected basic charge variant. And that does have a mass shift of 128 Daltons. So we can pick that up pretty well. We have good resolution. And we're actually able to get to quantitate levels of C-terminal lysine reasonably well. But overall, the main thing that I guess going into the method development that I was maybe skeptical of or concerned with was the sensitivity of using a mass spec approach for some of our critical glycans, in particular MAN5. Our expected specification for several of our biosimilar products is going to be less than 1%. So for a relative abundance of less than 1%, we weren't sure how reliable or how precise or sensitive this method would be compared to a fluorescence-based method, which we have a long track record with that, and very well characterized. I would say the initial results were very good. We can go through some of the data later, but we were able to reliably pick up these high mannose peaks, which was encouraging to see. This next slide has the light chain and again, the FD, or the fab portion of the heavy chain. And these peaks were not glycosylated, so they're a little bit more simple to look at. But we do have oxidation on the heavy chain. So if you look at like the innovator, we don't see any pre-peaks on that. What's labeled here as peak 2, or relatively low amounts. What I suspect these peaks are, so if you look at probably can't see my cursor, but if you look at the bottom trace, with the very bottom left trace, we've got a strong pre-peak on peak 2. We expect those to be methionine oxidation. So we knew from the sequence that there was a methionine 34 and a methionine 105, both of which are critical in this particular case because they, in the CDR domain, and so they will impact binding. But because we are doing a fabricated digest, we don't have the site-specific details on where that oxidation event occurred. So what we were able to determine was looking at gross changes in oxidation, which for our purposes in clone selection and clone screening is actually sufficient. We don't need to go down to the site-specific level of, is it the MET 105 or MET 34 that's oxidized? We just know if we're seeing, gross increases in oxidation, then that's a concern. But for our purposes, we don't need to characterize what each of these peaks are specifically, and so we didn't look at that in detail. So we know that there was oxidation taking place, but we don't know specifically what this pre-peak and post-peak are. Here's the results of the relative abundance for our primary glycoforms of interest. So on the left, we have the, results on the top half, and then each of the fragments with a C-terminal lysine are in the bottom half. All of these results aligned very well with what we saw by both ICIF and the fluorescence-based methods for glycan quantitation. And down to, we were able to go down less than 1% and see good agreement between the methods, which again, for the sensitivity of this method was a surprise to me, and a very pleasant surprise, because we're able to look at both our primary charge variants, with the C-terminal lysine, as well as quantitate the relative abundance of our primary glycoforms of interest reliably across 3 different samples with fairly different glycan profiles. So overall, very encouraging to see for me. The light chain glycation, heavy chain glycation, and oxidation were also reported. Again, we don't have site-specific information given the digest that was done, but looking at gross changes, this actually is kind of all we need for the early clone selection during cell line development, and as well as for kind of initial upstream media optimization. So this was good to see. We were excited. This figure restates the data in a visual form, so you can kind of see the variation of the relative abundance for innovator. I call vessel 1 and vessel 2 here. So there are 2 different clones, 2 different media conditions. So we do see variability, but it was expected. So that was good to see that we were able to pick up those differences reliably. And then you can see the relatively low level or relative abundance. So MAN8, we have way less than 1% for 2 of our samples. We were able to detect that using this method. MAN5 is what we used for our kind of critical quality attribute for our clone selection. So that's what we honed on more, but we were able to detect glycoforms even below 0.5%. So this was good. And then lastly, on the method performance, this can visualize the accuracy and reproducibility. So these samples were sent to 2 different sites within Waters and run by the same method on different instrumentation. And those results are shown here on the left on this bar chart. Overall, the method reproducibility was very good. Here we expect all of the bars to be the same height. And by and large, there were some minor differences for some of our lower level glycoforms, but generally very pleased with these results. And for the intended application of this method for Similis, more than sufficient method reproducibility. And then on the right, we have the percent difference from the released end glycan, the Waters wrap fluorescence-based method, and the relative variation was minimal. So the largest we saw was less than 3%, and even for our low-level glycans like MAN5, we were seeing 1.5, maxed out at 10%, but typically less than 2 to 3%. We're seeing very small relative differences between the fluorescence-based method and the mass spec-based method. So again, for our intended application, this was great to see. And so we kind of showed that these results, this method and these results were reliable, accurate, and reproducible so that they could be used for decision-making at our stage of development. So that's the method overall. Kind of summarize, our goal was to have a highly scalable method. We're looking at a lot of different samples for a lot of different cell lines across 20 to 30 different products. We're doing a high number of transfections. We have a lot of RCP, so automation and scalability in sample preparation was very important for us. We needed to be able to analyze clarified cell harvest. So again, this kind of comes back to the scalable sample preparation process, and run this alongside drug product. So we were able to achieve both of those, and then we needed method sensitivity that was sufficient for our intended application, which was clone selection. And the primary quality attributes we were looking at were glycan profile, honing in on the high mannose, MAN5 glycoforms because of the implications for PK. And then the C-terminal lysine variants was another point of interest because it is our largest expected variation across cell lines in terms of charge heterogeneity. So it's kind of an intrinsic attribute of the clone or the cell line that if we can start from a good place, then as we move into upstream development, we need to do fewer modifications with trace metals or temperature shifts or kind of tweaking to get that charge profile to align with the innovator. So we were able to, and I guess we were able to demonstrate that we could do both of these quality attributes, everything that we needed for our clone selection and early process or early upstream development sample testing with one scalable method. So yeah, it was exciting results. And that is overview of the method. Happy to take questions. I guess a big thank you to Sam, the whole Waters team. They did all of the heavy lifting, so well done. You surprised us on the quality of the method, both in terms of sensitivity, accuracy, reproducibility. So very well done. And then from the Simula side, Brad and Dane were also involved. So big thank you to them. So maybe that was quick, I don't know, but happy to take questions, or I guess Nick, if you want to, yeah, I guess kind of guide from here.
Yeah, sure. If we want to move on to the next slide. Thanks very much for that, Jared. Really nice talk. And don't worry, not too quick at all. We like to keep these things kind of punchy and sharp. So that's absolutely perfect. And really nice to see that comparison data with the other techniques as well, and that lining up so nicely. So yeah, kind of underlines what we try and promote as being the benefits of a system like the BioAccord. So thanks very much for sharing that. I'm going to go on to, I've just got a slide here, which is a bit of a plug, but before I do this, I think we're going to ask a very quick poll question. So we always like to give the audience the opportunity to get the appropriate follow-up on anything that they've seen in terms of the technology or any kind of specific questions. So the poll question that we are asking here is, would you like more information about the Waters products and solutions that you've seen here? Would you like to be contacted by a Waters representative? And the options are obviously either yes, please contact me, or no, maybe you're not quite ready for that conversation yet, which is also, of course, absolutely fine. So we'll leave this poll question up for just a moment or two and let people answer that. And Julia, I'll ask you to take that down once enough people have had the opportunity to answer. Just while that poll question is up, though, I would like to tell you about another event that is upcoming from Waters, and that is an event that we call WebAF 2023. Now, this is the Waters Biopharma Analysis Forum. It's gone virtual, so the E this year stands for electronic. We've gone virtual. We want to open up this event to as many people as possible. And we've got some really, really nice talks lined up already from our scientific community. So WebAF is very much a scientific community event. And the speakers this year are going to include Immunocore, GSK, Bayer, and the universities of Geneva and Strasbourg. So if you're interested in this, please do scan the QR code. Just to note that we're going to move slides in a second, but I've also put the QR code on the next slide. So there's plenty of time to scan that. That will allow you to find out more, and I hope you are able to join us there. And also do look out for some nice other biosimilar related content from Waters in the coming months as well. Okay. I see Julia has shared a link in the chat as well. So thanks very much for that, Julia. Great. If we could move on to the next slide, please.
Oh.
Perfect. So you can see here the QR code on this slide is for the WebAF event. So please do scan that and take a look at that webpage. So we've had a few questions coming in throughout your talk, Jared, and we'll go into those now. So I'm just going to open these up and I'll read them in sequence. The first question we have was, were you able to quantify the relative abundance of C-terminal lysine in the samples tested?
Yeah, we were. I didn't share the quantitative results because they're, I guess, a little bit more difficult to interpret relative to what we considered the baseline truth, which was the ICIF data. So ICIF, you have your basic 1 and basic 2, and so those correlate to one appended lysine versus 2 on your 1 heavy chain or 2 heavy chain. And so if we get a result of 18% C-terminal lysine, that doesn't directly correspond to how much percent basic 1 and basic 2 that we see. That being said, we could kind of look at those results in aggregate, and they aligned really well. So for example, for the vessel 1, I think we were seeing around 18 percent by ICIF, and then using this method, we were seeing around 17% total C-terminal lysine in aggregate. And again, that, parsing that into basic 1 and basic 2, you can't do with this method, but for the purposes of what we, decision-making information we need, that information is sufficient.
So I guess in the context that you're using this data, is it more the trend than the absolute value that's of interest?
Yeah, very much so. I don't know if probably doesn't make sense to go back to like the ICIF trace, but looking at percent acidic and basic are going to be, you want to be in the ballpark so that when we move into upstream development, as we do our, media optimization, media supplementation, to really kind of fine tune those quality attributes. We know that we're going to be able to get there. That if you remember from the first slide, we're looking at like a CHO versus a SP20 cell line. We're seeing dramatic differences. So, like 30% basic to, you could see for like a CHO-ZN, maybe less than 2% basic. And so if you're starting from that place, then you won't, we don't expect to be able to kind of bridge the gap to get all the way to a perfect alignment with the innovator. But if we're in the ballpark, then we kind of have an idea of what we can do in upstream to modulate and, tweak those attributes a little bit and kind of know that we're going to be able to be biosimilar.
Absolutely. Cool. Thank you. The next question I've got here, is there the potential to speed up the analysis time by shortening the chromatographic method? I guess people were looking maybe at the elution time of the peak compared to the full length of the method. Is there potential to do that? Is there value to do that?
Yeah, no, I think it's possible to speed it up. I guess for the development exercise that we're going, 15 minutes for the samples that we're testing was sufficient. With the sample preparation setup, I guess that's not the bottleneck for the current method. So that wasn't where the focus was placed. But there's no doubt that 15-minute chromatographic method could be shortened. Whether it could be got down to like 2 minutes or 4 minutes. Some of those Pro A methods you get super short. I don't think that's feasible, but yeah, there would be some opportunities and potential to speed that up.
I think it's a good point. No, it's that question of where's the value in that compared to the total time of sample preparation and analysis as well. So yeah, it's a good point. Cool. Okay. Next question. Were you able to quantify other common post-translational modifications, such as N-terminal deamidation?
We were. So for the samples tested, there was significant levels of pyroglutamine. The difficulty there again is because the cleavage was done with Fabricator. We don't have site-specific information. So the like pyrrolization of glutamine, I think is, if I remember correctly, a -8 Dalton shift. But then methionine oxidation is a +16. And so you have multiple potential modifications that could be happening at the same time. And so it makes those quantitation of some of those common post-translational modifications difficult. As I mentioned, if you see gross shifts in oxidation, then we kind of believe we can trust that. And again, that's kind of correlated with the shift in retention time as well. But some of the, I guess, lower level post-translational modifications, because it's, dealing with really big chunks, can't really get too quantitative with that. But for us in the intended application of the method, that's not a problem. So yeah, we'll pick up gross changes and we'll have, we're not too worried about low level site specific stuff at this stage.
Sure. I guess that kind of ties in with the next question that I'm going to ask then about the context of the data. But the 0.5% reporting cutoff, was that driven by the performance of the method, or could you have gone lower if you wanted?
We could have gone lower. It's a difficult question to answer with a statistical backing, just because we don't have or I guess I don't have the length of time with this method to make any like statistical claims about intermediate precision, or what's the, what's the limit of detection, or the, in any quantitative way. So I guess most of the reporting decisions were based off of kind of qualitative assessment of variability across the samples tested. And, that 0.5%, I think we felt comfortable with that. Going lower, do you see signal? Yes. Is it how, even the precision at, like some of those really small peaks, if you do like a 10 microliter injection load by the fluorescence-based method, down at, 0.1%, you see high variability there. You really have to increase the injection load quite a bit to get good precision on some of those really small peaks. And so some of it, without getting too far into the weeds, is it possible to go lower? I think, yes. What's the expected precision of, if we go down to 0.1, 0.2%? It's difficult to say. We need to run the method quite a bit more and kind of get a statistical history to make any claims about that.
Absolutely. No, that makes a lot of sense. And yeah, just because theoretically it's possible to go lower doesn't necessarily mean that data is valuable. So
yeah, and I guess for our application, if we go below, there's not really a big incentive to push much lower than that. Like for example, the PK implications of the high mannose, we think we'll have a specification of, less than 2% with targets of less than 1%. But, at the end of the day, the impact there is those high mannose glycoforms are bound by mannose receptors on your macrophages, and so they're cleared more quickly. But, if you have less than 1% of your glycoforms, which are cleared at an accelerated rate, in like the, what is it, B phase of your clearance, like it doesn't have a huge impact in vivo. So, the overall expectation for product potency is minimal. So, going down to the 0.1%, is it scientifically interesting? I think so, but in terms of kind of decision making for what we're after, I guess there's not a big driver to push down to super low levels right now.
Absolutely. It's very, again, contextual as to the decision you need to make as well. So
yeah, definitely.
Cool. Okay. Well, thank you. I think that's all the questions that we've got. I'm not seeing any more coming in, so I think in that case we'll probably draw this session to a close. Just want to say thank you again, Jared, for sharing those perspectives with us. Really, really nice talk, and as I say, this will be available on demand as well. Thank you to everyone that's joined us at any point during the whole series. We appreciate you taking your time, spending that with us, and learning a little bit more about some of the Waters solutions. Thanks to all our other hosts, our presenters. I do want to put a big shout out to Julia and Sean. They work very, very hard behind the scenes to help deliver these series. So thank you very much to the two of you as well. We've had 4 great episodes, as you can see on the screen here for the series. As I mentioned, those recordings will be hitting your inbox shortly, so please do look out for those. If you've got any further questions, you can see there's a couple of email addresses on this slide here. Please reach out anytime using those contact details. More than happy to help. But otherwise, I think all that remains is for me to say thank you again for joining us, and hopefully we'll see you all at another Waters event in the very near future. So thank you.